Okay, so we have some leftovers from last week. I didn't show you how to use gene regulatory network analysis. So this time I'm going to need you go through that analysis, and then I will leave the rest of time for yourself. Either you want to share your screen to present your analysis, or if you have any difficulty during the process, let me know so I can give you the hand-on guidance, and then we work it out if you have any problems. So last time we do have the problem with the gene regulatory analysis, and with the example, I didn't be able to show you the results, but this time trying to use example data again. So example data here, so I load this and still use the scatter plot of single cell experiment object created. Okay, so this is my example data. If I want to build a gene regulatory network for one of the group of cells, first I want to highlight, see this is a group blue cells, so-called master cell, and then this is a pyramid of cells. And master cell. So for instance, I'm going to build a gene regulatory network for this data set. The button I'm going to click is this called build gene regulatory network. Okay, click this. Now I can build a network for all the genes. So this data set contains 9,830 genes. If you can see my cursor here. So this algorithm will allow you to build this network contain all the genes. However, this is time consuming, and for this small data sample, we really want to select a few genes. For example, if you select a couple of genes using this multi-gene selector panel, so I just randomly select a few genes, but if you have an idea that what gene you should select, should be selected. For example, gene involved in the oxidation of the this process, you may want to group those genes because at the end, the gene regulatory network you build will make more sense to you because you know what kind of a molecular process you are working with. But for now, I just use this example, random set of genes to be included in my gene regulatory network to show you the example. So once I finish select the genes, maybe a few more. Okay, I can see click done. Now the program is working on to build a regulatory network. That's the gene expression correlation between genes. Okay, this is O by O. So each gene I selected will be included here, and to compute its relationship regarding with its gene expression profile across cells. And if, for example, these two genes always co-appeared in the same cells, and there's positive correlation between these genes expression, there's a new correlation will be built and highlighted in the blue. Okay, And if the coefficient is higher, the absolute value is larger, then the link will be stronger. And you see the thicker links here. And if the relationship is negative, that means if this gene expression level is high, another gene expression will be lower. Then the link will be in red. Okay. So that's the network we found here. And this is O by O. And even though there's the link between these two genes, for example, is very weak, we still draw a line between them. However, you can use a filter to filter out. So you change this cutoff to filter out. See, I only keep, if you only keep 5% of top edges, you see like a 95. Here, I want to keep 15% of all the edges between genes, which is strong enough, and I will get rid of those weakly link. I can see like 85. Then this will clean up the messy network structure here, not O by O, but only the most important ones are left. So you can see these two genes, RAB20 and ITP-KB, they positively correlate to each other. Their direction is upregulate this may upregulate that as well. And this gene negatively correlated with a lot of other genes. Okay, so you start to look at their relationship across genes. So this gene set I randomly selected, it doesn't make much sense to me because I don't know those genes. But if you select right gene regulatory module, you may know exactly why from the other evidence, previous knowledge, for example, this gene should negatively correlated with this because they are supposed to be anti-correlated. Okay, their regulatory relationship in these cells to be negative correlation. So this is, by the way, this is a build from for all the cells. Okay, I haven't got a chance to select which cell type I want to build this. So this network is for all cells combined. Once you have this network, you can say change something like this weight and change weight. Yeah, this is for illustration purpose. If you want to emphasize, you see, when I click this, the weight becomes stronger. Just visually, it's bolder lines now. And you can increase the size of the gene a little bit. Okay. And this is change the layout. I'm going to click this later. And this one will switch this so-called directional graph. So you see this each linker between genes, there's arrows. That means the information is from this gene to that gene and not the other way around. The other way around was here, but it's too weak. Has been filtered out by this cutoff. So this graph is called directional. It tells you the regulation relationship and who's upstream, who's downstream, and who regulates who. However, a lot of analysis does not require this. You can change this to unidirectional. Okay, so if I click this, now the errors are gone. I still keep 85%. Then you see, okay, there's only undirected graph in here. Okay, give me a break here. Yeah, so there's a way you can switch between directed and undirected. So any questions so far for this kind of analysis? Yeah, so I'm going to change the layout a little bit. So if you decide to click this button, you can see, okay, you want to rearrange the genes. These genes with no link to any other gene. So they are out of this connected module. And there are different ways to show this. Click. Switch between different kinds of okay. So there's animation you can make. Okay, so to show you, so when you start to cut off, increase the level cut off, then gradually you trim off those weaker links. Okay, so the last one, that's the highest, strongest correlation between genes. All right. So this is the one analysis you can build for your gene set. In order to build the cell type specific ones, for example, I want to build a network for this yellow genes only. So I can highlight, for example, what this gene, the cell type, okay? Pyramidal cells number 5. So I can click this button, which is select cell by class. Cell type. I want to select number 5. All right, so this allows you to select a particular group of cells. Okay, so from that plot here, I extract this particular cell type out as an independent sample. Now I click this button. I can select genes as what I did and build a network. And this network will be for this particular cell type only, right? So in this way, you can build cell type specific network. I hope it makes sense for you right now. The next question is I want to compare the network I construct from this cell type against that. So in order to construct two regulatory network and compare, you may want to use this tool to compare two single cell gene regulatory network. I click this, I will select which group with two different cell types. So I will collect, this is called the master cell number 3 cluster against pyramidal cells number 5. So select these two. I will compare these two groups. So select OK. And now again, we need to select genes. So you can click this button to have that dialog in order to select genes. And also I provide you something like this. You can paste your gene names. So here are just some random gene examples. And you can type your genes in here. So we are going to build a network for those genes and compare between two samples. Two samples means one is from this cluster of cell, another from this cluster. So you can compare the difference in their gene regulatory networks between two cell types. So once I click OK, the algorithm is working. Now you have two networks. Okay, on your left should be the master cell 3. On your right side should be this pyramidal cell number 5. Okay, we will be able to do the same thing to trim off or change. But this time we'll be apply these actions to both networks at the same time. Okay, by looking at these two, and you can immediately have your first impression is in cell type number 2 and this PLCB gene. Looks like there are a lot of interaction or co-expression pattern with many other genes where this gene was not so much involved in any of this. So that's our first impression. And there's another revarying event around this DHRS7B, right? In this sample, there's no connections with other genes, but here you have a lot. So this gene has not much change, but it's become weaker a little bit. And this gene also changed a lot. So if you found this information interesting enough, you may want to go to Google this gene. For example, what this PTP4A3 gene is doing. So you may Google gene card information. So this gene function phosphorylase. Okay, signaling, right? There's a process. But anyway, so this becomes so weak in this cell type. It was stronger here. And then you can search other genes as well. Okay, so this is reductase, another enzyme. Okay, so in this way, once you have enough comparison and you have enough data sets for the gene, significant genes, then you will work out the change of these two regulatory networks into different cell types. By the way, this gene highlighted in red are transcription factors, which we know they are regulators in many molecular cell process. So they are highlighted. Okay. Yeah, so that's all the analysis we can do with the cells in this single cell scatter application. And this is a two network related studies. And for differential expression gene, and if you compare this set against that set, I think this is one of the button which one can use is compared to between two classes. So you want to find this gene's expression difference between two groups, for example. So anyway, so this button will show you the difference. So this gene's expression level in this 5 different clusters look, this purple looks expression level higher than the other group. So it's not so clear for this particular gene, but if you test different genes, you will see the difference. And also this one, if you compare two groups, For example, master 3 against 5. So you select two groups. This will start to compute which set of gene and differentially expressed between these two groups. Okay, so here you have a table to show you, okay, this gene expression level is higher in one group and lower in another group. There are significant values, there are fold change. So that's a report you may want to use to find the gene differentially expressed across samples. For example, remember this name of gene, so this AF. So this gene, so we can take a look. What's the difference between this gene between two groups? If you look at this single gene expression level highlight, and then this ARFIP2, and you can see this gene has been identified as a top gene, which expression level is quite different between this group and that group. If you highlight this gene's expression, that's indeed, because it's highly expressed in this gene set, but relatively low in other gene set. Okay. So this table will help you to find those genes through the differential expression analysis. Okay, so that's all I'm going to share, and I'm going to stop the recording and give you time to practice and then ask a question. Okay.
I have a quick question.
Yeah.
So, with looking up what the individual genes do, like the ARFIP2 gene, would looking up what that does in comparison to other ones help with our analysis at all? Or is just knowing like the cell type and relationships and prevalence all good?
Yeah, so you can Google, certainly search the function of this gene. Once you have a couple of these kind of genes, you start to figure out what this cell type is. Because this gene only expressed in high in this cell type. So this is a differential expression analysis is slightly different from the network analysis. But you certainly can combine those information you learned from this exercise and then try to figure out the possible underlying principle about these gene activities. If you want to try another, this could be COX-8A, so single gene. COX-8. So this gene, you see, same pattern, and it's highly expressed here. Okay. Let's fold change. Let's find a fold change which is positive. So those fold changes negative indicate the second cell group have a high expression. So they all show same pattern. So I'm going to look for a positive value in this fold change. So this one, for example, if I search Jamin2, this gene's expression. You see the opposite pattern here, right? It's highly expressed in here, but not in that. Okay, any quick questions?
Also, for the recordings of the previous classes,
Mhm.
is there a way that you can like email those out to us or put them on eCampus just for reference for some of the students that missed the meetings?
Yes, so I do have this uh, Zoom, and also I put on the YouTube. I will send you a link from YouTube. That's okay?
Okay, thank you.
Yeah. Yeah, so I'm going to stop recording this because the rest of time it will be yours. If anyone want to share your screen to let me know the problem you have, I will be able to help you online here.